This method can help answer key questions in insect surveillance and control by improving the attraction of existing traps and incorporating colors or lights into the design of future monitoring and control traps. The main advantage of this technique is the initial evaluation of attractive colors and insect behaviors in the lab prior to the costly and time-consuming tests in the field. Insects have innate behaviors or instincts that should be used to make monitoring and trapping more effective. The Bioassay arena is designed to be a flexible method to evaluate behaviors in any insect species prior to field trials.
This protocol uses an aerial arena, but a terrestrial arena design is also available. The critical difference between the aerial and terrestrial arenas is the use of vertical space, which changes the size of the arena. When selecting an arena type for an experiment, consider the insect species being studied.
The aerial arena has the ability to capture insects in a collection container, whereas the terrestrial arena is a two-dimensional object and really is designed for recording visits to the lights.
To begin, disassemble and clean the aerial arena with an odorless, non-abrasive soap and warm water. Then dry the arena and allow it to finish air drying. Before each experiment, record the pertinent environmental conditions and monitor any trends that occur from experiment to experiment. First, use 4 supports and a black linen cloth to set up a pedestal. Then place the arena base plate